Good evening, everyone, and welcome to the class on experimental biochemistry, which is being taught by Professor Shagata Das Gupta and Professor Shomodaya of IIT Kharagpur. I am Srijan Sen, and today I will be taking the live session of week 9. Like I always do, I will be discussing in brief first the lecture slides, and that has been already that you people have probably seen or gone through, and then we will be discussing a few question and answers. Okay, and that will help you in probably solving the assignments and all. Okay, so I will just wait for a few more minutes, and then we can actually start the class. Okay, I'm sharing my slides with you all. You can confirm me. So let's wait for a few more minutes. Okay, and then we can actually start with the class. Okay, so let us start. If anyone else comes, we can add them later. Okay. So in today's class, we will be mostly discussing about enzyme. We last class we talked about enzyme kinetics. In today's class, we will be talking about isolation and characterization of DNA. So let us start from the very basics. So we have all heard about biomolecules and so biological molecules and all. So these are divided into four different classes. One includes the carbohydrates or the sugars, like you have the glucose, the fructose, galactose. There are so many different types of um, and different carbons. Having different carbons, sugars, like you have the two carbon, the three carbons, you have four, five, six, it can go up to seven. Okay, so depending on the number of carbons and the structure of all of these sugar molecules, you give it different names, like you have glucose, then you have fructose, you have galactose. Okay, one of the category is carbohydrates, the other one is lipids. So lipids are molecules which we also know them as fats, and these fats have so lipids have a hydrophilic tail that remains on the outer surface for interacting with the water, the hydrophilic head, and the hydrophobic tails that remains on the inside of the membrane and all for giving it a rigid like structure. So the hydrophilic head comprises of choline, then you have glycerol, phosphate, so phosphoglyceraldehydes. You have so many different names that are given to you have the phospholipids, the and so many different types of lipid molecules that are present over there. So just you can remember that the lipid has a hydrophobic tail and a hydrophilic head. If you see over here, so you see the heads are mostly towards the outer side of the that interacts with the water, because they're hydrophilic in nature, they're water loving. So okay, and then you have the hydrophobic tail that forms the inside of the portion, so it does not interact with the water molecules, and it forms the basic structure of a you see you have the phospholipid bilayer. Okay, the PLP bilayer. The third category is proteins. So proteins, you have heard of that. So one of them is an enzyme. So proteins are three-dimensional molecules that are formed by alpha amino acids, and they have a particular structure and function. So the third category is your proteins. The fourth one that we are mostly interested in today's class is nucleic acids. So nucleic acids comprise of basically the DNA, the RNA. Okay, and so nucleic acids are mainly comprised of sugars. You have a phosphate group and a base. So you see these are the sugar molecules. You have the phosphate group over there that's attached. Okay, so this is the glucose. From glucose you have derived the structure and all. Okay, so you have the then you have the pentose sugar. So this is the pentose sugar. Okay, and you have the phosphate group attached over here. This is the base. Okay, that we are considering. And this is your double-stranded DNA that you're observing over here. So it has a, you see it forms different, so these dotted are the different hydrogen bonds that are formed between the bases. Okay, so nucleic acids are mainly comprised of sugar molecules. You have the phosphate base, the phosphate group, and the base, right? So what are nucleic acids? So nucleic acids are materials that store and transmit hereditary information. It stores the information that is present in a genetic code. On the other hand, RNA carries the information. So RNA, you can tell that it transcribes the information from the DNA, and it's translated or it's carried forward to the protein. That's the synthesizing machinery for carrying out the different types of functions inside the body or an organism. So the structure of the DNA and the RNA, as I was saying, it has three major components. One is the phosphate group. You have the pentose sugar, and the other one, that is the nitrogenous base. Now, depending on the nitrogenous base, it's allotted, the molecules are given different names. The phosphate group, the pentose sugar, remains same. Only the thing that varies for a DNA or RNA, there is a few differences between a DNA and a RNA that I will be talking about. But more or less, the components includes the phosphate group. You have this pentose sugar and the nitrogenous base. So the phosphate group, you see the structure is constant over here. Now for our deoxyribose, so what is the DNA? So deoxyribonucleic acid, and ribose is ribonucleic acid. So deoxy, so that means the 2' OH that is present is replaced with a hydrogen over this. So it's deoxyribose. So that's the ribose sugar, and it contains 5 carbon Cs. That is why it's a pentose sugar. It's pentacarbon. Now the difference between DNA and RNA is that the 2' OH is present in an RNA, whereas for a DNA it's the 2' hydrogen. So the OH is missing and we have the hydrogen over here. Okay. So what's the difference between them? The first difference that I mentioned over here is the OH and the hydrogen of the 2' in the 2' position. So this is the ultimate structure. So now the nitrogenous bases, you can categorize them actually. These nitrogenous bases can be categorized into pyrimidines and purines. So these are the nitrogenous bases, the structure of the nitrogenous bases. So you see over here, so in the pyrimidines you have cytosine, thymine, uracil. So you see the basic structure is the same for the pyrimidines. Okay, the only place where it varies, if you see for a cytosine, let me show. So you see you have N C N C double bond C and C. So you see for a pyrimidine, all of this is basically the same. So this is actually maintained. Okay, the only place where you see it actually varies is the position over here. You see it varies. For thymine and uracil it's same, but for a cytosine it's different. The other thing that you will observe over here is you see this double bond present over here. That's another thing for a cytosine. Okay, and then you have for a thymine the position. This position actually differs. So the basic, if you remember the basic structure of pyrimidine, that's fine. Then you have to remember in which position what was present on that. Similarly for purines also, you see the basic configuration is the same, and then you will see minor changes over here and there, like you have in this carbon the position over here. Okay, and you see it's C double bond O. See this position. Then the position that is over here, you have the hydrogen, and over here you have the NH2. So that's the difference between an adenine and guanine. So what you need to remember is definitely the structures is what you need to remember. Like for a cytosine, what's the structure? Then for a thymine or a uracil. Then also you need to remember that adenine and guanine is under purines, and cytosine and cytosine, thymine, and uracil is for pyrimidines and purines. Okay, so you have purines and pyrimidines. In pyrimidines, you have cytosine, thymine, and uracil. In purines, you have adenine and guanine. Okay, so this is the structure of the DNA and the RNA. So as the unit, these three components make up a monomer. Now, what's a monomer? So it's an individual unit that acts as a building block for large biological molecules. So monomers are nearly the identical units. Now these monomers combine with each other to form large molecules that are made up of these small kinds of monomers in the form of a chain, giving rise to a polymeric structure. Like you see over here, 1, 2, and 3 are the monomeric units, and this is attached by a particular chain to form the long polymers. Now these polymers can be formed by the formation of a bond between the different monomers, that can be by dehydration, removal of water, or different other ions. Just the information of a bond can be enough for the formation of a polymeric unit. Okay, so monomers are basically the smallest unit that are involved. That is the basic unit of a polymer, and polymers can be you have a longer chain of polymer. This can be branched polymers, unbranched polymers. There are different types of polymers that can be able to form. Okay, now in the presence of water, be it an acidic medium or something, it breaks down into small monomeric units, and then you get different the polymers just break down by hydrolysis. That's the addition of water molecule, breaking of a bond, and you get the single monomeric units. Now you see over here that for a DNA or an RNA, what's present over here. So you see, this is for a thymine. Okay, so this is one particular monomeric unit. Now, for as you remember, in the DNA you had a 3' OH over here, and for a phosphate group you have an OH. I'm considering you have an OH. So the hydrogen from over here and the OH over here is basically removed, and you get the chain or the formation of a bond, right? So addition of a thymine monomeric unit and an adenine monomeric unit gives rise to the thymine adenine, and in this way you get basically a long chain of polymers, which we call as a DNA strand. So this is the DNA nucleotide. So nucleotide is the one that comprises of phosphate, sugars, and nitrogenous bases, right? Okay, so the hydrogen over here, and the OH over here is basically removed. So removal of one water molecule leads to the formation of a bond between the two monomeric nitrogenous base or nucleotides, and this is basically your sugar phosphate backbone that maintains the uh, structure of our DNA, because this is the responsible for the backbone of our DNA. The places where it actually varies is the bases. So you have a thymine over here, you have adenine, cytosine, and the guanine. And then when another strand comes, then there is the formation of hydrogen bonds between the nitrogenous bases, and depending on that you get a double stranded DNA. Okay, now there is an so the double stranded DNA gives rise to another term known as base pairing. So base pairing is a term from where we can understand that two bases are joining up, but how do they join? That is via the formation of an hydrogen bond. Like if you see for a thymine, so thymine forms a bond with adenine, guanine forms a bond with cytosine. But how many number of hydrogen bonds are there? So you see that thymine adenine is two hydrogen bonds, and for guanine cytosine is three hydrogen bonds. So the more the number of guanines or cytosines or the GC content in your DNA strand, the more stable will be your DNA, because it will be difficult to break the it is you have to break more number of hydrogen bonds to actually separate them out. So higher the GC content, higher will be the stability or the melting temperature of DNA. So what you need to keep in mind is that thymine pairs up with adenine and guanine pairs up with cytosine, and the number of hydrogen bonds between the pairs is that Ta is 2 hydrogen bonds and GC is 3 hydrogen bonds, right? Now you see that this is a double stranded DNA. Okay, so by the formation of, so one thing goes from, so this is basically your phosphate group, and this is basically your 3' region. So this one is your 3', this one is your 5'. So if one strand moves from 5' direction to the 3' direction, the reverse, this is your reverse strand that moves from the 3' to the 5' direction, or in an opposite manner, 5' to 3' direction. So in this manner, you get a double-stranded DNA where the proper base pairing is very, very important. So that's the double helix. Now this shows two strands of DNA in the form in which the DNA is present within the cells. Okay, so the distance between one loop region, that is in one turn you have 3.4 nanometers that comprises of 10 base pairs. So the pitch, okay, so the pitch of our DNA is equal to 0.34 nanometers, because this is for 10 base pairs. So for each the difference between the each base pair is basically the pitch of an DNA, that is about 0.34 nanometers. Now, nucleic acid strands. So as I was telling, that one strand moves from the 5' direction to the 3' direction, the other one moves from the 3' direction to the 5' direction, right? So nucleic acids are polymers derived from nucleotides. The backbone consists of repetitive units of phosphate deoxyribose units. Then you have the base that varies over this. So individual nucleotides are joined by phosphodiester linkages. Like proteins, nucleic acid chains have a polarity which is defined by the 5' and 3' ends of the sequence. By convention, you write the sequences of bases in a strand that is written from left to right, that is the 5' to the 3' end. So what does it mean? So you have two strands which we write like this. The first one is your 5' direction to the 3', because over here you have the phosphate group and here you have the OH, but the reverse strand is 3' to 5', and this is the complementary strand. So this is your forward strand and this is your reverse strand. Okay, and you see there are so many multiple number of phosphodiester bonds. Okay, phosphodiester bonds you see. Right, so the structure of a double helical DNA. So the cellular DNA exists primarily in a right-handed double helical form. So the double helix consists two interwound antiparallel DNA strands. The strands are complementary and paired together via Watson-Crick base pairs AT-GC, and the backbones of these strands are located to the outside of the helix where the bases are stacked inside, and you will see that depending on the stacking or the formation of the double helix, there are two different grooves that are actually formed. So you see there is a smaller groove over here and there is a larger groove over here. So depending on the cavity that's being formed, you call that has a bigger groove, you call it's the major groove, and with that has a that's of lesser width, you call it the minor groove. Now in the most common conformation of a double helix, that's your BDNA. So you have the ADNA, BDNA, ZDNA. Okay. So the common one is the BDNA. So 10 base pairs occurs per turn, as I was saying, and each 10 base pairs have a distance of 3.4 nanometers. Therefore, the difference between the two nucleotides is the pitch of the DNA, that is 0.34 nanometers. And DNA molecules contains wider grooves called the major groove and narrower grooves known as the minor grooves, in which parts of bases are exposed to the outside. DNA binding proteins locate and interact with specific base sequences exposed in these groups, and those proteins that interact with the DNA, if you know the term, it's known as the transcription factors. So transcription factors are those proteins that interact with DNA or nucleic acid and carry out different regulatory functions involved in our body. Now, a very interesting term is DNA bending. So, what is it like? DNA can be bent because it does lack stabilizing bonds oriented parallel to the axis of the double helix. And bending mostly occurs on binding of transcription factors, like I was telling, for example, the Tata box binding protein, the TBP, that binds to the TATA region, and the DNA binding proteins such as histones. It's crucial for packing of the DNA in chromatin inside the eukaryotic cells. Now you will see that in many cases multimeric proteins achieves different large sizes, about 10s to 100s subunits. So you see you have the DNA over here, there are several transcription factors, then you have the RNA polymerase, the mediator complex. So all of them comes together, your DNA ultimately bends to accommodate all of these proteins, and then you get your original protein or the original function is actually expressed. Now, such complexes exhibit a higher level of structural organization known as a supramolecular structure. Like you have the mRNA transcription pre-initiation complex, ribosomes, proteosomes, spliceosomes. Okay. Now, typically, supramolecular complexes function as macromolecular machines in reference to the fact that the activities of individual subunits are coordinated in the performance of some overall task, like protein synthesis by a ribosome. The base catalyzed hydrolysis of RNA. So DNA and RNA differ with respect to the sugar found in the nucleotide monomer units. The phosphodiester bonds DNA and RNA differ with respect to the sugar found in their nucleotide monomer units, like the 2-deoxyribose versus the ribose. The phosphodiester bonds of RNA are susceptible to hydrolysis in basic solution due to the presence of 2-hydroxyl group of ribose. In contrast, the phosphodiester bonds in RNA are much less susceptible because of 2-deoxyribose that lacks in this group. Now, it is thought that DNA was selected over RNA as the preferred molecule for long-term storage of information because it is a less reactive molecule due to its containing the 2-deoxyribose. Now, DNA denaturation is a term that refers to the unwinding and separation of the two strands of double helical DNA molecule. In vivo enzymes such as helicases, for example, separate DNA strands. In vitro denaturation is achieved by heating or by the treatment with low ionic strength buffers or extremes of pH. DNA denaturation can be monitored by UV absorption spectroscopy because GC base pairs are held together by 3 hydrogen bonds, whereas AT base pairs are connected by the 2 hydrogen bonds. The melting point of the double helical DNA increases with G plus C content. In DNA renaturation, double helical molecules are formed by the annealing of single-stranded molecules. DNA supercoiling, so what is that? It's a process such as the replication that winds the double helical DNA to introduce the torsional stress that results in supercoiling. It is most evident in circular DNA molecules such as the bacterial plasmids and some viruses, but it occurs in linear eukaryotic chromosomes as well. Replication causes overwinding ahead of the strand separation site as positive supercoiling. Now, underwinding can occur resulting in negative supercoiling. So, these enzymes are called topoisomerases that regulate the amount of supercoiling in DNA in vivo. Topoisomerase 1 relaxes supercoiled DNA. Topoisomerase 2 introduces negative supercoils into DNA, and the cellular DNA naturally exists in a slightly negatively supercoiled state. And negative supercoiling facilitates processes such as replication and transcription where DNA is unwound. Then you have the RNA structure, which is known to most of you. So most cellular RNAs consist of a single strand. However, double helical regions are common in RNA, where complementary sequence region occur. Like the common types of RNA secondary structure elements, you have the hairpin, the stem loops, then you have the pseudoknot because of the complementary sequences. And they also form elaborate three-dimensional structures in molecules such as the tRNA and the rRNA. The pseudoknot tertiary structure found in human telomerase RNA. The 3D structures of ribosomes rival those of proteins with respect to the complexity. Now, what do we need DNA for? We need it to detect, enumerate clone genes, and then detect enumerate species to identify the different DNA regions for recombination. Okay, so what about RNA? So which genes are being transcribed, when and where are these genes transcribed, and what's the level of transcription? But before that, we need to understand the DNA purification. So you have a culture of bacteria that is grown and harvested. You centrifuge it, you get a pellet of cells. Now the cells are removed and broken to give a cell extract. So you have the cell extract over here. You go for cell harvest and lysis, and then you have to purify the DNA and concentrate the DNA to get the DNA concentration. So what happens over here is that, so how do you prepare the cell extract? So you have the cells over there, you disrupt the cell wall via some kinds of harsh reagents like SDS, or even basic mediums like NaOH that rips apart the cell wall, and the entire materials comes outside where you have the DNA, or the supercoiled DNA, that's the plasmid in the case of a bacteria. So you use detergents, organic solvents, lysozymes, heat. So you can use many of these things. Then you centrifuge to remove the cell debris and the chromosomal DNA. If you need, that's fine. If you need the plasmid DNA, that's also okay. So you collect the supernatant and purify it. Also, there are ways of preparing the genomic DNA. So this is mostly for plasmid DNA, but for a genomic DNA, there are many ways. So you have the cell extract, you mix with phenol. So you will see there is a separate layers by centrifugation. So you will have a phenol, you will have the interface with coagulated proteins, and your aqueous layer will have both the DNA and the RNA. Then you can collect it and add maybe RNase A to remove all the RNA molecules, and from there you can just collect the DNA molecules. But you need to be very careful such that you do not affect the genomic DNA and break them ways. So there are two ways of concentrating the genomic DNA. One is via spooling, the other one is ethanol precipitation, where you are adding chilled ethanol and your DNA gets precipitated. You centrifuge it, you collect it and mix it with a solvent, maybe a buffer, and then you check the concentration and all. For the genomic DNA preparation in plants, and the other thing that happens over here is that there is carbohydrates that are present which you need to remove. So you have the cell extract, you treat it with cetyl tetraaminobutyls. Okay, that's a cationic detergent. You centrifuge it, you have the carbohydrates and the proteins in the solution that is removed. So you will have the nucleic acid CTAP complex. You discard the supernatant, you resuspend the pellet in 1 molar of NaCl, and then again go for an ethanol precipitation ribonuclease treatment to remove the complex and get the pure form of DNA. Okay. So the other way of, okay, so what are plasmids? So plasmids are vehicles for recombinant DNA. So you have the genomic. So in a bacterial cell, you have the genomic DNA and you have the plasmids. So plasmids are external materials that you put inside a bacteria to maybe express your protein or something. So that's the non-chromosomal DNA, and it undergoes replication that's independent of the chromosome. Many copies can be carried per cell. It's easy to isolate and manipulate. So you can do it for recombination of proteins. Then you have the plasmid purification via alkaline lysis. So you have the linear DNA, you see, you treat it with some alkaline medium conditions to denature the DNA, and then you neutralize it. So genomic DNA cannot renature that fast. So plasmids can, because they're never fully separate. So there is a tangled mass of linear DNA coiled over there. So it's a very haphazard or a very messed up situation. So it ideally it pellets down. Okay, you take the supercoiled DNA, that's a plasmid. You take the supernatant and load it into a column where it can bind the DNA. Then you elute it using a buffer or even water for doing that. So like I was telling, so other columns are usually made up of silica particles. You pass it through the cell extract, the supernatant basically, through these kinds of a column, and your DNA just binds over there. Then you elute it with water, you get the DNA. Then you have, for a DNA purification, you have the phenol chloroform extraction, where you can use 1:1 phenol chloroform, or the other one is 25:24:1 phenol chloroform isoamyl alcohol. So what is the role of the different agents? That is very important. So phenol denatures the proteins precipitates form at the interface between the aqueous and the organic layer. Chloroform, it increases the density of the organic layer, and isoamyl alcohol, it prevents foaming. So this is the slide that I just mentioned. So what happens with RNA? So the problem with RNA is that RNA is chemically unstable. So spontaneous cleavage of phosphodiester backbone via intramolecular transesterification, and it's susceptible to nearly ubiquitous RNA degrading enzymes, RNases. RNases are released upon cell lysis. It's present on the skin, and it's very difficult to inactivate. So disulfide bridges conferring stability, no requirement for divalent cations for activity. So what is PCR? So polymerase chain reaction. So it's first described in 1971 by Klepp and Corona, and then it was redescribed in the year of 1985. It allows the amplification of specific sequences that have defined endpoints. It's a fast, powerful, adaptable process, and it's also very simple, and it has a lot of applications. Like, why do we need it? So it's usually used for cloning, for recombination studies, for sequencing, other in vitro studies. You can identify the engineered DNA constructs, like I was telling, for recombination of DNA, to monitor the gene expression, to diagnose a genetic disease, to reveal the presence of a microorganism, identify individuals and all. So what do you basically need for PCR? You need to know first the sequence of the template, that is your target sequence that you want to amplify. You will need some short oligonucleotides that will define the ends, the region which you want to actually amplify. So this is very important. This primer should be short, as well as should be able to complement with your DNA, such that they can sit over there, and the DNA polymerase can actually put the DNTPs with the help of the buffer composition, that are the activators, and increase the length of the DNA and amplify the sequence. So how does it work? So a simple PCR reaction, you see. So this is a double-stranded DNA over here, and you denature them with the formation of heat, followed by primer annealing. So you, at very high temperature, so at high temperature usually the proteins break, but these are the DNA polymerase is thermostable, so it can accept up to a very high temperature. So you first open up the double-stranded DNA, you allow the primers to sit and start annealing. Then the DNA synthesis, the primer extension stage, where the DNA polymerase just extends via the addition of the DNTPs. And then your DNA strand amplifies. So the cycles of denaturation, you have primer annealing, primer extension by the DNA polymerase, and you carry out this. So this is for the first cycle. First is, so remember, the first stage is DNA denaturation, then comes primer annealing, and the next one is primer extension. Okay, so this is for the first cycle. There will be multiple cycles, and in each cycle you get two strands of DNA. So after 30 cycles you will get 2 to the power 30. That is about a huge number, and that's the number of DNA you get at the end of a particular number of cycle. That's 30, let's assume. Okay, and your DNA ultimately amplifies. So that's about the polymerase chain reaction. Okay, now we will be talking, discussing the practice problems. 2 to 3 minutes of break, and then we can start with the practice problems. So now we will talk about the practice problems. Okay. So to lyse the bacterial cell wall for extraction of DNA, which of the following reagents is commonly used? You have lysozyme, phenol, penicillin, and C-tab. Yes, that's correct. You have lysozyme. Okay. Then question 2. So isolation of DNA from plant sources poses challenges due to the presence of high amount of RNA, high amount of protein, presence of polysaccharide and secondary metabolites, low amount of DNA. So why is it difficult to isolate DNA from plant sources? Is it because of RNA, proteins, DNA, polysaccharides, metabolites? What? Carbohydrates, polysaccharides, secondary metabolites? Yes. So option C is correct. That's good. Now, the function of EDTA during cell lysis. So what is the function of EDTA? What's the function of EDTA during cell lysis? It acts as a metal chelating agent. Yes, that is correct. Inhibits cellular enzymes to degrade the DNA, destabilize the cell wall. So all the three options are correct. So option D, all the above. And next one, what is the function of NaOH in lysis buffer? I was just telling about it. Denatured genomic and plasmid DNA, break down the cell wall, degrades the RNA, remove proteins from the cell lysate. What is the role of NaOH? Yeah, it does not degrade the RNA. Okay, it denatures genomic and plasmid DNA and it breaks down the cell wall. So A and B both are correct. Question 5. RNase A degrades RNA that are present in the cell lysate. What will happen if RNase A treatment is not done during DNA isolation? So what will happen if you are not adding RNase A during isolation of DNA? So what will happen? So DNA gets degraded, RNA will not be removed, and as both DNA and RNA absorbs at 260, it will be difficult for the quantification. Both DNA and RNA will form complex and precipitate out of the solution, or all of the above. It's a very interesting question. Yeah. So if you're not adding RNase A, then the DNA does not get degraded. Rather, RNA will not be removed, and both DNA and RNA will absorb at 260. So that will become problematic in quantification, and that is the correct option. It does not precipitate out. Okay, let's move on to the next question. Question 6. What's the role of SDS in lysis buffer for plasmid DNA isolation? Solubilizes the membrane, denatures the proteins, disrupts DNA protein complexes, all of the above. So DNA is a detergent So it solubilizes the membranes, that is correct. It also denatures the proteins and it disrupts the DNA protein complexes. So all of the above are correct. SDS is a detergent, so it rips apart the membrane by solubilizing it. Fine, because the membranes will be all negatively charged and there will be a charge repulsion. The membrane just opens up, it gets all the materials from the inside, just gets out. Then the SDS reacts with the proteins over there and it denatures all the proteins. We all know it how it does. So repulsion effect. And then when the proteins gets actually denatured, it disrupts the DNA protein complexes, and what you get is only the DNA. Okay. The next one. What's the purpose of plasmid isolation? Why do we need to isolate plasmids? Cloning, gene expression, driving DNA, target DNA into other organisms, or all of the above. So from just I was speaking about the applications of plasmid isolation. So when I told you all the options would be correct. You can do it for cloning, gene expression, identification of the DNA in other organisms. Then if you want to check for a disease or so you can do all of this recombination and also all of the above is correct. Okay. Another one. Isolating plasmid DNA from genomic DNA. So how do you isolate plasmid DNA from genomic DNA? Based on conformation, charge difference, size difference, cellular localization. So how different is a plasmid DNA from a genomic DNA? Let's see, what do people think? Okay, yeah. So based on conformation, that is correct. So conformationally, plasmid DNA is different from genomic DNA. There is no charge difference. The size difference is there, so that's correct, A and C. And the cellular localization is also not correct because for a bacterial cell, you see it's present in the genomic DNA or the plasmid DNA remains together. Now, so in plasmid, during plasmid isolation, we use a neutralization buffer. What is the composition of this neutralization buffer? Phyton X, potassium sodium acetate, SDS, phenol. Yes, that is also correct. You have the potassium and the sodium acetate because you are adding an alkaline solution to it, that is an NaOH. So for neutralizing it, you are adding an acetate to neutralize the pH. And what happens when you're neutralizing it? The genomic DNA cannot get back to its original conformation and it ultimately gets into the pellet, whereas for the plasmid DNA it does not get hampered a lot, so it coils up and you get it in the supernatant. Now when neutralizing buffer is added after lysis to the cell lysate, so what happens? Oh, just I mentioned it, so it's another easy question. So chromosomal DNA gets tangled and precipitates out. Plasmid DNA anneals and remains in solution. Proteins and SDS forms insoluble precipitate, all of the above. So just as I was mentioning, so the chromosomal DNA does get tangled and it precipitates out. You have the plasmid DNA in the solution because it can anneal itself. The chromosomal DNA being very large in size, it cannot coil up, and the proteins and the SDS remains as insoluble precipitate. So all of these is the correct option. The next one, that's the DNA isolation when you are adding that supernet into the column. So it's a question related to that. DNA bound to silica membrane can be eluted out by the addition of. So what do you need to add to elute out the DNA out of the silica membrane or the silica column? Is it water, isopropanol, tris of pH 6, or an SDS solution? So for elution, you can definitely use water. Okay, that's correct. And then, but the other options are not correct. So isopropanol you can use for washing of the DNA, for the precipitation of the DNA. Okay, you cannot use an SDS or a tris of pH 6, right? Okay, the next one, to wash DNA bound to the silica membrane, which of the following reagents is used? Tris, deionized water, ethanol, isopropanol, or detergents. So for washing, you can use ethanol or isopropanol that you can use for washing out the DNA. That is also 70% ethanol, not 100%. And the others cannot be used for washing out of DNA. Right, so option C, that's ethanol and isopropanol. So remember, for elution you use water, for washing you are using ethanol or isopropanol. Okay, and we get back to the last question. That's in agarose gel electrophoresis, the plasmid DNA runs ahead of the linear DNA. Why? Linear DNA is more compact. Plasmids have higher mass by charge ratio. Plasmids are supercoiled and migrates faster, or all of the above. So why does the plasmid DNA run ahead of the linear DNA? So linear DNA is more compact. That is wrong. This is wrong. Plasmids have higher mass by charge ratio. That is also not correct. For all respects, plasmids are supercoiled and migrates faster. That is the only correct option over here. Okay. So that's all from my side. Thank you for joining. If you have any questions, you can ask me. If not, then we will again meet in the next week. Okay, so no questions. Thank you then. We will again meet in the next week. Okay. Thank you very much for joining.